When we think about what has changed in the last 5 years of understanding human evolution, there is no story that is bigger than the story of the Denisovans. This is a population that we know today only through their genomes. And it's a population that archaeologists had not suspected they would find to exist. It's a really crazy story, and it's one that so illustrates the importance of ancient DNA in reshaping our view of the dynamics and interactions of ancient humans in the past. We have, through ancient DNA technology, recovered evidence of a population that is among the ancestors of living humans today. And that's tremendously exciting. But it also shows us just how much the fossil record and the archaeological record leave out as we're considering the dynamics of ancient populations and how they came to be modern humans. Denisova Cave is a cave in the Altai Mountains of Siberia. It's a region that is today in the nation of Russia. And the Altai Mountains are themselves a very interesting area. There's archaeological evidence of very early modern humans, of Neanderthals, and now of this third population, the Denisovans, all of which were existing in what is a relatively small area near the borders of Russia, Kazakhstan, Mongolia, China. It's a tremendously interesting area, an area that is really the crossroads of Central Asia. And it's an area that has been investigated intensively by Russian archaeologists for now, at Denisova Cave, more than 30 years. The Denisova excavation has produced really interesting evidence of the behaviors of ancient people. And until recently, everyone assumed that these ancient people that were being documented there were likely Neanderthals. I will say that at Denisova Cave itself, there are bones that do represent Neanderthals because we have ancient DNA evidence from some of them that's very similar to Neanderthal specimens that are found elsewhere in Europe and in Central Asia. But the evidence from one of these bones was very different from Neanderthals and even more different from most living people. That evidence came from one bone, which is the tip of a fifth finger, or a pinky, and represents an older juvenile or young adolescent female. We know it's female from the 2 X chromosomes in the genome. That bone produced better, more well-preserved ancient DNA evidence than any other bone had before this one was sequenced. Denisova is truly an extraordinary situation in terms of ancient DNA preservation. The reason why is that the Denisova Cave is a cold cave year-round. It's a cave where the average temperature around the year is 0 degrees Celsius, so essentially freezing. That low temperature, maintained over tens of thousands of years, has effectively preserved DNA from multiple specimens. So the really, really interesting genome came from a pinky bone. It's something that, as a paleoanthropologist, I have to say is shocking. This is the kind of bone that, if you're really a specialist, you will know that there was a pinky bone from Denisova Cave. But otherwise, you're not going to pay a lot of attention to it. The pinky doesn't tell us a lot about the behaviors of ancient hominins. You could probably do your dissertation on every pinky that was found anywhere in the world, but it wouldn't be a very interesting dissertation. Osteologically, this is not great evidence about this ancient group, but the genetics give us the 3 billion base pairs copied twice that existed in this ancient person's genome. They're telling us today far more about this ancient population than we could ever have discovered from bones alone. So it's hugely important. How do we know the Denisova individual represents a different population? When we look at the sequence of this genome compared to human sequences, compared to Neanderthal sequences, it is very different from both of them. Now, across the genome as a whole, it's slightly more similar to Neanderthals than it is to living people around the world, and especially to living people in Africa now. So there is some relationship between this genome and Neanderthals, but that genome is very slight compared to the relationships among modern humans now, and it's very slight compared to the relationships among different Neanderthal sequences that we have. So this genome appears to represent a group that was quite distinct from Neanderthals and quite distinct from recent humans. It was a different population. We know something about the dynamics of that population. For one thing, this genome shares some functional genes with Neanderthals that lived in Central Asia. Those functional genes are genes related to immunity. And so there was interbreeding between this population and the nearby Neanderthal populations, interbreeding that was sufficient to spread at least some genes that had adaptive value. We also know from this genome that it has parts that are very divergent from Neanderthals and from recent people. Most of the genome is sort of somewhat divergent, but some parts of the genome are very divergent, and that includes the mitochondrial DNA. The mitochondrial DNA of the Denisova genome is very different from Neanderthal mitochondrial DNA and very different from the DNA of living people. Living humans, for their mitochondrial DNA, share an ancestor around 200,000 years ago. We share an ancestor with Neanderthal mitochondrial DNA around about 500 to 700,000 years ago. We share a common ancestor with the Denisova mitochondrial DNA well more than 1 million years ago. Coupled with evidence from other genes that also show this pattern, the Denisova genome looks like it represents an interaction with a population that was yet more different from us than the Neanderthals were. The Denisova genome may have interbred with something like a Homo erectus population or a significantly divergent archaic human population that we haven't yet discovered. That is so interesting because it's showing us the dynamics of populations that we can't witness with bones alone. We also know something about the population in which the Denisova genome existed. That population was smaller than the population that gave rise to living people because the Denisova genome is more inbred than living people are. So Denisovans were a pretty tight-knit group, a group that was relatively small and persisted for a long time, relatively quite separate from other populations, but with some interbreeding with quite distant populations. The other thing that we know about the dynamics of the Denisova population is that they interbred with the ancestors of some living humans. We know that because when we compare the Denisova genome to the genomes of people living today in Aboriginal Australians, Highland New Guinea, to some extent Melanesia, which includes New Caledonia, New Britain, and nearby islands, and in the very easternmost parts of Indonesia and across the Pacific in Polynesia, when we compare the genomes of those people, we discover that they have similarities with the Denisovan genome that no one else in the world today shares. So the Denisovans were partial ancestors of the genetics of these populations that live at the southeasternmost extreme of the old world human existence. That is super interesting because what it tells us is that the Denisovan population was extensive enough that the emerging population of modern humans, carrying with them a very high fraction of African ancestry, interacted with and interbred with this Denisovan population, giving rise to the colonization of Australia, of Melanesia, and of other points in the Pacific, and that later there was a movement of people that did not carry the Denisovan signature of intermixture that spread across Asia and covered up the evidence of this earlier mixture. Present-day Asians have a very, very small degree of Denisovan admixture. In Aboriginal Australians and in Highland New Guinea, we see 5% of the genetic heritage of those people is a population that was like the Denisova genome. On the mainland of Asia, it's well under 1%, maybe as low as 0.2%. So we're looking at a very complex dynamic in which human populations emerged, they mixed, they separated to some degree, further populations emerged and mixed and colonized new areas. Later populations emerged and spread and covered up the evidence of some of the earlier mixture. Human populations in their genetic history are what archaeologists would call a palimpsest. A palimpsest is a manuscript that was written on and then rubbed out and written on again. In medieval times, monks that were copying manuscripts sometimes reused the same pages. Modern human genetic ancestry is like that. Our ancestry comes from multiple layers of ancient movements that emerged from Africa, became somewhat differentiated from each other, mixed with each other, and then remixed as later people left Africa. It is a really complicated and interesting scenario, and the Denisova genome demonstrates it more than anything else. But the Denisova genome's not alone. We're increasingly learning, as we'll see later in the course, that when we look at ancient DNA within regions of the world, we discover that the ancient DNA comes from populations that no longer are strong representatives of the ancestors of living people in the same regions. Ancient Europeans that lived before the time that agriculture spread into Europe represent only a small fraction of the ancestry of living Europeans. Ancient Indonesians that were there as Australians originated from that area are no longer representative of present-day Indonesians that lived in the same area. And the Denisovans show that that goes back into deeper time periods, so that modern humans are coming from a complex process where ancient variations are being established and then remixing, and then partially being rubbed out by the movements of new people that are expanding with some mixture and successive layers of this process. The Denisova genome is telling us about these ancient dynamics. They're dynamics that go well back into the Pleistocene and that involve archaic humans that lived in Africa and that lived across Eurasia. It's a tremendously interesting discovery. And there are other aspects to it that involve the functions of these genes. I'll talk about the importance of gene functions and the way that we're learning about the biology of these ancient people when I talk about Neanderthal genetics. Because in those cases, we're able to discover something about the biology of an ancient population beyond its bones. And the Denisova genome is giving rise to similar insights about a population where the bones that we have are very, very few.